Hello everyone and welcome to today's Keynote webinar titled Function First Live Cell Screening and Microwells for Application and Cell Line Development. This webinar is a part of the 9th Annual Laboratory Automation Virtual Event Series. I am Melanie Chan of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located on the left of your screen and follow the process to obtain your credits. Now, let's get started. I now present today's speaker, Benjamin Yellen, co-founder and CEO of Celldom. For a complete biography on our speaker, please find it on the left of your screen. Benjamin, you may now begin your presentation.
Thank you for the kind introduction. Good morning, everyone, or good evening, depending on where you are. My name is Ben Yellen, and I am the CEO of Celldom. Today, I would like to give you an overview of the work that we are doing with microwells for applications in cell line development. So I thought it would be helpful to first start off with a brief background on the history of techniques for developing monoclonal cell lines. The first and oldest approach is known as limiting dilution. This approach involves adjusting the cell concentration and the aliquots so that 0 or 1 cell is deposited into each well of a plate. This method is certainly the easiest, but the problem is that only 20% or fewer of the wells receive a single cell, while the rest are empty. Therefore, this approach doesn't make good use of the plate real estate. Furthermore, since the cells are isolated in a large volume, they don't receive the growth factors and extracellular signals from other cells, which causes them to expand less quickly or not expand at all. As a result, very few clones can be grown out in each plate, and they are being selected based on their ability to survive in isolation rather than the desired functional property. To solve the first part of the problem of low sorting efficiency, the next generation of techniques have made use of flow sorters to establish 1 cell per well initial conditions. However, the disadvantage of this approach is that it introduces additional stress, which causes fewer of the cells, at least on a proportional basis, to expand. Additionally, it doesn't solve the problem of that cells are being selected based on their ability to survive in isolation in a large bath of media without any neighbors around. So these issues have motivated the development of new techniques that can perform function-first screening of cells prior to isolating and expanding them into cell lines. In my view, these emerging approaches can be classified into 3 categories. The first is microwell technologies, which capture cells by gravity and constrain their positions with nanoliter chambers. The second approach is droplet technology, which partitions cells in nanoliter water droplets surrounded by oil. And the third is microfluidic technologies, which involve continuous perfusion of cells trapped in nanoliter chambers, which are sometimes called pens. So let's take a closer look at each of these emerging technologies. Of these 3 classes, microwell technologies are the simplest and the most flexible. They have the advantage of being able to perform many continuous measurements of the same single cells over time, which is needed to determine growth rates or other functional properties, like its ability to secrete antibodies or growth factors at a high rate, or its status of differentiation in the case of iPSCs, or its ability to develop novel reporter cell lines from various types of monoclonal sources. Another advantage of microwells is that the cells are physically isolated from the cells in other microwells, but they're exposed to a common media, which helps the cells grow better. Finally, of the 3 emerging technologies, microwells are unique in their ability to handle complex samples, such as patient biopsies, organoids, and co-cultures without clogging. The main disadvantage of microwells is the stochasticity in cell loading, which is driven by Poisson statistics and therefore leads to many microwells that are either empty or have 2 or more cells. However, these microwells can be identified through time-lapse imaging and discarded. Therefore, the main symptom is that only 1/3 of the microwells will have a single cell. However, in our case, this means that we still have 100,000 microwells with single cells in each experiment. Another popular class of emerging technologies is based on droplets, particles, nanovials, nanoshells, or other suspended solid or liquid supports that can encapsulate single cells and sort them in a microfluidic chip or in a conventional flow sorter. The main advantages of droplet technologies are that the empty droplets can in some cases be sorted out, that's if the cells are stained with a fluorescent marker, and it can be low cost. However, the disadvantages are that it is difficult to track the same droplet over time, and therefore you only get a single imaging time point for taking these functional cell measurements. Secondly, droplet generators have well-known clogging issues which make it more difficult to handle complex samples like patient biopsies. Finally, due to the constrained volume and inability to remove waste products, these cells can't expand significantly inside of droplets surrounded by oil. This can be potentially solved by nanovial, nanoshell, and other types of approaches. However, they also have their own different set of limitations. The final class of emerging technologies is perfusion-based microfluidics. These platforms typically involve introducing a suspension of cells into a microfluidic device and then moving the cells into different nanochambers based on optical, electrical, magnetic, hydrodynamic, and other types of forces, after which the cells can be cultured for many days and quantified by various types of fluorescent readouts. The main advantage of microfluidic platforms is that cell loading is better controlled. And this is particularly important in the case where you may want to pair two different types of single cells in each chamber with high precision. Since media is continually being refreshed in these systems, these cells can also expand on chip, and they can be monitored continuously by time-lapse imaging. However, the problem with these platforms is that they tend to be very expensive and complicated. They require constant perfusion, and they also have clogging and fouling issues that prevent either the introduction of or retrieval of specific cells of interest. Based on this competitive analysis, at Celldom we chose to develop our platform based on microwells, which we believe is the best solution for cell line development, and that's what I'll focus on for the rest of my talk today. The platform that we have developed for cell line development is called the Clone Explorer. This instrument is the combination of a live cell analyzer, incubator, laminar flow hood, and clone picker all in one. It can take transmitted light bright field images and fluorescent images in 4 channels. It has onboard temperature, gas, and humidity control, which allow us to perform experiments for weeks or longer without affecting cell viability. A typical experiment for us lasts about 7 days, during which we perform real-time monitoring of cell growth rates, surface markers, cytokine secretions, and other cell properties, which allow us to identify which cells need to be retrieved and converted into cell lines at the end of the experiment. The consumable in our platform is the CloneX plate, which is a Microwells inside wells format, and it is structured as a 96-well plate so that we can test many samples or treatments in parallel. Finally, we have our distiller software, which compiles the time-lapse imaging data and allows for selection of the final pick list. So let's give you a quick view on the guts of the instrument. On the left, we have the cell culture plate, which is being imaged by inverted light microscopy. The microscope is underneath, it's not shown here. On the right, there is the cell deposition plate, which is temperature controlled, and this serves the purpose of adjusting the rate of evaporation in the deposition plate and also to cool the sample where necessary. And in the middle, we have a wash tray, which is used to sanitize the tip in between each clone picking event. And then there is the cell picker. This moves in between the 3 plates, picking cells from the cell culture plate, depositing it into the cell deposition plate, and then washing it into the wash tray, and then repeating that cycle. So when we were setting out to validate the Clone Explorer platform, we focused on a few key metrics that we believe are the most relevant to cell line development. First, we needed to show that we could grow cells in microwells, and that the growth metrics were similar to what would be observed in standard cell culture incubators using multi-titer plates, and then that the growth rates were also free from edge effects. so that cells could be picked from any well of the plate and have the same robust response and expansion. Having established viable growth conditions, next we needed to show that the cells with the desired functional properties could be consistently identified, and this required us to establish compatibility with fluorescent measurements, including surface markers, secreted cytokines, as well as bright field measurements of morphology, growth characteristics, and also cell-cell interactions like cell killing, which I'm not going to talk about today. Since our plates have 270,000 imageable microwells, we needed to develop high-throughput bioinformatic pipelines that use advanced computer vision tools, which can analyze the contents of each microwell, and the analysis speed had to be at least as fast as the image acquisition speed so that all of the images could be analyzed by the time of picking. We also needed to validate that our picking method did not damage the cells and therefore affect their viability, and also that picking did not introduce contamination either from between adjacent picks and also other sorts of contamination. And finally, we needed to show that the method can quantify the memory of a clone, which means we are asking the question of whether the cells that are expanded are the same as the cell properties that they were selected on. So let's take a look at each of these one by one. So our first goal was to establish the appropriate incubation conditions where cell lines grow really well. On the left, we show an example of one well. This is the E2 well in a 96 well plate. And in this well, we loaded 3,000 leukemia cells, of which we ended up with 1,052 microwells with a single cell out of 2,869 imaged microwells in that well. This coincides with the expected distribution of single cells from Poisson statistics. In the middle, we show an example trajectory of a single cell, which is overlaid with the best fitting exponential growth, theoretical growth trajectory. Within 4 days, a typical microwell that starts off with a single cell will have between 10 and 20 cells. And so the mean division rate, which is what is being measured for each trajectory, which is gamma in this equation, is obtained and then plotted as a distribution for each well on the plate. Fortunately, we found that cells grow uniformly across the plate without any edge effects. In other words, the cell in the A1 corner well grows as fast as the cells growing in the center of the plate. This means that colonies can be retrieved from any well on the plate without growth rate artifacts. Having demonstrated the ability to consistently grow cell lines, we also aim to establish that proliferating cells could be recovered from primary samples. Since T cells are often the ones used in cell-based medicines, we aim to assess whether our platform could be useful as a characterization method to assess changes in the clonal makeup that occur during various phases of the cell manufacturing process. To address this question, we plated donor T cells in different types of activation matrices, media formulations, and growth factors such as IL-2, IL-7, and IL-15, aiming to understand the conditions that lead to robust growth and also maintain the diversity of the original sample. This is one clear example where single-cell analysis yields insights that would be difficult to obtain from a bulk measurement. Specifically, here we compared Thermo Fisher Dynabeads and Miltenyi Transact CD3-CD28 T-cell stimulation reagents. And we found little difference on the clonal structure of the expanding clones based on these two different activation media. So it is just not true that every cell will expand. And in fact, we found that only 30 to 50% of clones were proliferating, and even fewer were rapidly proliferating. We found that the proliferating fraction depends mostly on the donor rather than the media conditions. And there was little effect on the CD8 makeup in the expanded population, regardless of the type of activation method. So ultimately, for our purposes, we showed that this platform can be used with more sensitive primary samples, but it does also open up a lot of questions on how to optimize cell manufacturing processes. So next, we aim to assess the functional properties of cells in each microwells. So for the functional measurements, our first goal was to show that our optical system has the sensitivity to classify populations based on their surface markers. So in our platform, we have a 12-bit camera, and this provides enough sensitivity to separate the CD4 and CD8 populations using 2 fluorescent channels. The other 2 fluorescent channels can be used to measure various properties, such as stemness, activation, as well as other functional markers. Our method for measuring surface markers is based on identifying cells in the bright field image, and then drawing a segmentation mask around those cells, and then measuring the mean fluorescent intensity within that mask in each channel, and then plotting that, such as that shown on the left. One of the advantages of working with our high aspect ratio microwells is that we can replace the media without disturbing the cells. So this actually makes it possible to perform more complicated assays, such as performing a live dead assay stain at the final time point, just by replacing the media and incubating it. So this showed us that we can measure properties of individual cells based on their fluorescent markers. Next, we aimed to extend this to measure products that are secreted by cells, such as cytokines or antibodies. First, we were interested in establishing the lower limits of sensitivity for secreted product detection inside microwells. For these measurements, we used spherical beads that were coated with cytokine capture antibodies, and we added known concentrations of interferon gamma to different wells, while then monitoring the mean fluorescent intensity of the beads in each microwell. So as you can see on the graph on the right, the signal is linear over 3 orders of magnitude, and it has a lower limit of detection approaching 10 picograms per mil, which is as sensitive as gold standard ELISA assays, and convinced us that we can measure cell secretions quite accurately and at the appropriate concentration levels. Okay, so having established the limits of detection, next we aim to show the ability to measure interferon gamma secreted from individual cells inside those microwells. In this experiment, we established a co-culture of dendritic cells and T cells from a HLA-0201 donor that were exposed to MART1 peptide, which is a peptide derived from melanoma cancer cells that is known to be overexpressed in the subpopulation. In these experiments, we opted to coat the walls of the microwells with the cytokine capture antibodies instead of using the beads like before, because we found that dendritic cells are very hungry and they have a tendency to eat the beads. And secondly, because the fluorescent signal that we measured had a characteristic ring shape that could be easily detected using a combination of computer vision models and statistical analysis. As you can see, the microwells have low background in the control population, which didn't receive the MART1 peptide, whereas the cells had a really robust response in the wells that received MART1. In effect, we have developed a real-time digital ELISpot assay that is incredibly easy to use. You just add cells, reagents, and image over several days, and then you get your results. But this is what we use as a precursor to our picking, which I'm going to show next. So, having shown that we can grow cells uniformly and robustly, and having established that we can measure functional properties of cells based on surface markers and also the volume of their secreted products, also called their titer, our next goal was to show that we can retrieve high priority clones based on the desired combination of growth rates and functional properties. Okay, so here's how the cell picking process works. There's a diagram on the left which shows the cell picker tip and how it fits into the microwell. So here, the conical shape of the microwell is very important because the tip is wider than the microwells at the top, but not at the bottom. So the constriction fit between the tip and the microwells protects the cells from damage during picking because the tip just physically cannot reach the bottom of the microwells. Moreover, the conical shape of the microwells also guides the tip into the microwell of interest. So these two properties in combination allow us to automate the picking process without requiring a high degree of precision in placement of the tip. So let's show an example of how this picking process works in action. As you just saw, first we take images at the first time point in bright field and multiple fluorescent channels so that we can validate the specific microwell that we aim to pick in our pick list. Then the picker moves into the field of view and lowers until it strikes the substrate. Once found, the tip is then moved into the microwell of interest, after which the contents of that microwell are retrieved, and then the cells are moved into the deposition plate, after which the tip is sanitized for the next pick, and then a post-pick analysis is done so that we can validate that the cells are retrieved from those microwells. So to assess the robustness of picking efficiency, we performed a picking test in which cells were loaded with fluorescent beads, and the instrument identified the microwells based on position and then retrieved beads from microwells going from left to right. So in this example, we found that 95% of picking events were successful, which is defined as the removal of a majority of beads from any individual microwell. And as you can see, we've basically vacuum cleaned from left to right all of the microwells on this screen. So now that we have all the mechanical and biological parts working together, the next step was to develop the computational architecture that allows us to crunch so much data and display it in graphical format so that we can establish the final pick list. So for example, as in this platform, consider that for a full plate scan in which we image 270,000 microwells at 4-hour intervals, we need to analyze over 1 million microwell images per day. So that rate corresponds to 20 microwells per second imaging speed. If we take it to the extreme and image the plate every minute, which is possible with certain imaging technologies, then we would need to be analyzing 5,000 microwells per second to match that imaging speed. So clearly, computational speed is paramount to enable identification and picking of the best clones. It also became clear to us that we needed a user interface that looks very much like a flow cytometer so that we can gate the cells of interest and go from a population of 270,000 down to a pickable number like 96. So that's what we did here. We have followed the tried and true method of developing gating software that looks like a flow cytometer. However, there are a few key differences that are worth noting. First of all, our data points are actually microwells instead of cells, and therefore they have more complicated data types. For example, a microwell may have different initial conditions, say a co-culture versus a monoculture, and it may even be more granular, like there are 2 cells of type A and 1 cell of type B. Similarly, microwells also have parameters related to secretion of certain cytokines, and which also occur at different time points. So finally, cell trajectories can change over time, and therefore growth rates and other parameters that are ensemble averages of a specific cell are not something that are typically measured on a classical flow cytometer. So we have many more types of data. However, the approach to filtering that data remains the same if it can be graphed. So what we do here is we plot different types of data and then gate it. That leads to a next graph with fewer clones, and then we continue gating it until we get to the population of interest. So in this experiment, for example, we are concerned with CD8 positive T cells that are secreting interferon gamma and are growing. So these 3 gates allow us to go from 226,991 microwells, which was the initial populations for these 80 wells, down to a pickable number here of 98. All right, so having validated that the ability to establish viable growth conditions, select cells based on desired functional properties, and isolating the clones of interest, the next logical question is whether we can expand these clones. So let's talk about that. To assess purity of the isolated cell populations, we started off with a simpler model, which used 2 different cell types. One expressed a red fluorescent protein, and the other expressed a green fluorescent protein, and these were co-cultured in each well of our CloneX plate. Due to the random distribution of cells, some microwells ended up with a single cell of either type, but some microwells had more than 1 cell and some were empty. So using image analysis, we focused our picking strategy on 2 conditions, namely identifying the microwells with monoclonal cell populations, and 2, determining which of these microwells have robust cell growth. Once these were identified, we then retrieved the cells in alternating format, first red, then green, then red, then green cells, so that we could assess different sanitation strategies, for the tip in between each pick, and also assess its effect on downstream expansion of this isolated cell populations. As expected, our results did not show any contamination due to carryover from one pick to the next, nor did we have any colonies where a microwell was misidentified, meaning it was actually a co-culture when we thought it was a monoculture. Furthermore, we successfully expanded 86 out of 88 of the attempted picks, which in our eyes was sufficient to demonstrate the gentle nature of this picking method. So in conclusion, we felt it was a pretty successful demonstration, and we have the ability to isolate the clones of interest without contamination. So in the final slides here, I will address the most important question in cell line development, which is, do the cells display the properties that they were selected on? In brief, our conclusion is that this question is more related to plasticity of cell memory than it is to any measurement artifact of a cell picking platform, at least in our hands. And this is a natural question to ask, since a cell population is a quantity that continually evolves in time. So in the next few slides, I'm going to show a few examples where memory is retained, and also a few examples where memory is not retained, as guidelines for how one would establish stable master bank of cell lines. So one of our personal interests here at Celldom is to understand why some drugs can completely eliminate a cell population, whereas other drugs cannot, leaving behind drug-resistant cells. Therefore, we thought it would be useful to identify drug-resistant cells in culture, isolate and further expand them in vitro, and then use these cell populations in various assays, such as to test cross-resistance with other therapies, and also to inspect the pro-survival pathways that are utilized by these drug-resistant cells. So we performed this experiment on MOLM13 leukemia cells that were selected based on two parameters. First, they started off as a single cell, and second, they continued to proliferate in a drug, dividing at least twice in 7 days. And we then retrieved several dozen of these clones and expanded each population for use in a rechallenge assay. Our expectation was that most of these expanded cells would remember how to resist a drug. This is the property upon which they were selected, and that we would see more than 50% of these clones growing out in the resultant population. However, as you can see here, our results showed that each of the clones behaved qualitatively similar to the parental line that never experienced the drug. In other words, these cells likely did not have any permanent, i.e. genetic, alterations that would allow them to maintain their drug-resistant state. Instead, it became clear that these cells were developing transient methods to resist a drug that perhaps were of epigenetic or transcriptional nature and origin. So this was a very clear state in which a functional property of a cell population was not remembered. And that is because the property was based on selective pressure. Once the pressure was removed, then these cells quickly resorted back to their natural state. That's the working hypothesis here, although we were expecting to actually have developed drug-resistant cell lines. As an alternative example, there are cases where phenotypic properties are preserved, and ironically, it's not the state of fluorescence of a cell, which also was found to be variable, but rather a morphological property that we found to be consistent after many rounds of cell division. For example, returning to our experiment in which we retrieved cells from a mixed cell population of red and green fluorescing cells, one of the things that we noticed in the GFP positive cells is that there were actually two types of morphologies in the cell population. One population consisted of cells that preferred to aggregate as clumps, and they move around in the microwells through collective migration, like clumps moving in each microwell. Whereas the other cell population consisted of dispersed cells that preferred to remain separate and move independently of its neighbors in the microwell. So we retrieved various examples of clones expressing these different morphologies. And the way we identified them was that some were brighter than others, and that's just because the cells were overlapping. But what we found is that this property was preserved for many generations of cell division, and now we have two different cell populations derived from an originally what was thought to be a monoclonal cell line. So as a final example of applications that we are working on in cell line development, we have done a lot of work on retrieving antigen-specific T-cell populations. such as the previous slides where I discussed our ability to identify T cells that become activated upon exposure to MART1 antigen. So here we were interested specifically in the CD8 positive T cell population and also the ones that secreted and proliferated inside the microwells. The proliferation was important because we wanted enough material both to do TCR sequencing and also to further expand these specific T cell populations for use in rechallenge assays. So we are still working on the sequencing aspect of the project, and we will be reporting these results in a future journal paper. So I hope that my talk today has convinced you that microwells have a number of very compelling applications in cell line development. The microwells within wells concept is a pooled arrayed live cell biology platform. It is pooled in the sense that each of the cells is exposed to the same media or perturbations as the other cells, so we can study the statistical properties of a population of cells, such as how do cells respond to a small molecule drug or an activation reagent or other stimulant. However, it is arrayed in the sense that each cell remains in its own microwell through the duration of the experiment and can be retrieved individually for further expansion or analysis. So the reason why we're so attracted to this platform is the numerous advantages it has for cell line development. First of all, it allows for single cell resolution and the ability to perform measurements on hundreds of thousands of cells per experiment. This allows us to select the absolute best clones. Secondly, it has the ability to measure many types of properties on the same microwells at the same time. We routinely are now measuring surface markers, growth rates, and cytokine secretion, as well as cell killing, immune synapse formation, and other sorts of morphological properties. I actually don't like to call this platform single cell analysis because in my mind, it's more high replicate biology. This is necessary if you want to identify extremely rare cells in a population that may be as low as 1 in a million. That means you need more than 1 million replicates to find that one cell. And that's what microwells are doing here. But it doesn't have to always be at that scale. And we can actually use relatively few cells to obtain a measurement, specifically when certain populations are very precious and rare. One well only uses 3,000 cells, and in many cases that could be sufficient for a specific screen of interest. More importantly, the reason why we devised the plate like this is so that we can fit more samples and more conditions in every plate. And this has served to be extremely important in our experiments so that we can put all of the controls on the same plate and allow us to identify true biological effects. And finally, the reason that we work with microwells is that it's very straightforward to isolate clones. You identify them in imaging, you go in with a picker, and you retrieve them. And then these cells could be used to create cell lines or to discover novel biological sequences. So in total, the value in our platform is that it provides better characterized clones, it leads to clones that expand better in bulk, and our platform is compatible with various types of cells, ranging from cell lines to organoids to primary samples, allowing it to be used in a variety of formats for cell biology at single cell resolution. So with that, I really appreciate your time in listening to this talk, and I'm really interested to hear your questions.